Hello everyone, and welcome and thank you for joining us today to our first webinar mini-series, A Beginner's Guide to Single-Cell Sequencing Technologies. This webinar mini-series webinar includes 3 parts. Today is the first part, which is entitled Introduction to Advanced Single-Cell Sequencing Technology. Today we have Dr. Katarina Pankova here. Hello, Dr. Pankova.
Hello.
Thank you for joining us today. And Dr. Pankova has received her PhD in neuroscience from Maximilian University of Munich. In Germany in 2017, she earned her PhD and she worked on the neural circuits underlying visual system. In 2018, she joined a life sciences and healthcare consulting company, and where she worked as a consultant. And in 2021, Dr. Pankova joined Single Remote Technologies, where she oversees single-cell sequencing products portfolio as a product manager. So yeah, Dr. Pankova, stage is yours. Thank you.
Thank you very much for the nice introduction. I'm very happy to be here and have this opportunity to introduce you all to the amazing world of the single-cell sequencing technologies. So my talk today will be structured around 3 different topics. First of all, I will give you a brief introduction onto single-cell sequencing. I will describe you what it is in detail and how it actually works and why it's important. In the second part of my talk, I will touch our technology and I will explain how it works and what kind of applications it is suitable for. And lastly, in the last part of my talk, I will talk about successfully published case studies that have used our technology to generate valuable data. So firstly, let me talk about what single-cell sequencing actually is and why it is actually important. So you might know that next generation sequencing technology, and especially the transcriptome analysis using RNA-seq, has advanced field tremendously. It provides you valuable information on gene expression across all genes studied. The problem arises if you work with samples that consist of mixed cells. So in such scenario, if you use traditional bulk sequencing, you end up with an average expression profile of the entire sample. So the bulk sequencing takes the whole sample and mixes all the cell types present in the sample together. So in the end, you cannot really distinguish between the individual cell types that you might have in your sample. And for this reason, the single-cell sequencing technologies have actually been developed to overcome this kind of obstacle. The single-cell sequencing technologies take as an input individual cells. So this next generation, so the NGS library is generated for each of these cells separately, and then it's pulled together onto the sequencer and processed as a single sample in a sequencing experiment. So single-cell sequencing allows you to get precise information on gene expression in each of the cell types that is present in your cell. So you basically you can in the end get information for each cell type separately. We in the field often use this metaphor or analogy between the smoothie and the bowl of fresh fruits. So the bulk sequencing you can compare to a glass of smoothie where you have different kind of fruits mixed together. So basically you might have banana, kiwi, and strawberry mixed all together. But if you want to look at the properties of the individual fruits, it's really difficult to distinguish them from the rest because they are all blended together. So this is what happens during bulk sequencing. Single-cell sequencing in this analogy would be a bowl of fresh fruits from which you can pick actual individual fruits and determine the properties of each of them individually. So this is the greatest benefit of the single-cell sequencing that is actually out there. It enables you to study the individual cells separately. Now, the application of single-cell sequencing is huge. You can apply it to multiple fields. Most predominantly to the fields that deal with samples with high heterogeneity. So this is cancer research, neuroscience, immunology, as well as the developmental biology and stem cell research. In particular, in cancer research, the single-cell sequencing enables you to study the intra-tumor variation of the cells that are present in the tumor, because the tumor does not consist of one single cell type, but it's a mixture of different types of cells with different kinds of functions. And it also enables you to study the interpatient variation of the tumors coming from the same tissues across multiple patients. Single-cell sequencing is also important for studying the tumor immune microenvironment. So in particular, if you're interested in the understanding the interplay between the immune system and the cancer cells, single-cell sequencing provides you valuable insights on the gene expression of the immune cells that might not be the predominant cell type in your tumor immune microenvironment. So single-cell sequencing has been named the breakthrough of the year in 2018 by the journal Science. And in 2019, it has been named the method of the year by the journal Nature Methods. The number of publications using single-cell sequencing has been increasing steadily over the past couple of years. And we believe that this trend will continue in the future until at some point the single-cell sequencing will become a common technique used routinely in a lab, such as, for instance, right now PCR is. So the single-cell sequencing has developed tremendously over the past 10 years or so. On this graph, you can see different types of techniques that have been used to generate single-cell sequencing data. So they can be grouped into two major groups, low throughput technologies and the high throughput technologies. The history of single-cell sequencing dates back to 2009. So in 2009, there was a study published by Tang and colleagues, and this study was a true single-cell sequencing study because it described gene expression in one single cell. So since that moment, the technologies have developed rapidly over the past couple of years, and right now we can study in a single experiment thousands or even tens of thousands of cells simultaneously. So when I will be talking about the single-cell sequencing and the methods and the technologies that we are at Singularity using for single-cell sequencing, I'm talking about high-throughput experiments, meaning that in these experiments, you can get the gene expression information from thousands or tens of thousands of cells simultaneously in a single experiment. So this is the kind of high-throughput single-cell sequencing I will focus on. Now, let me briefly introduce to you our technology and explain how we actually achieve this high throughput in our single-cell sequencing experiments. So the core of our technology is called a scope chip. This chip is shown on the upper left side of the image. Basically, the chip is a simple handheld device that you can operate without any special equipment, just with a 20 microliter pipette and stuff that you already have in your lab. So on this chip, in this transparent part, there are thousands of microvels. A picture of them is shown on the right side. So what you do with this microchip is that you inject single-cell suspension into the microchip, and the single cells fall into the individual microvels gently by gravity, and they get partitioned. So we often use this term partitioning in the single-cell sequencing experiments, meaning that these single cells occupy individual compartments and they're separated from each other. So after cell partitioning, we load barcoding beads onto the microchip. And the size of the microvels and the barcoding beads ensures that only one barcoding bead falls into each microvel. So in the end, you end up in a situation where you have single cells and single barcoding beads in the individual microvels. Now, the barcoding beads themselves carry oligonucleotides on their surface. I will explain on the next slide what exactly is in those sequences. But what is important is that once the single cell and the barcoding bead come together, we lyse the cells release their contents, including the mRNA, and the mRNA gets captured onto the barcoding beads. So once this happens, we extract the barcoding beads out of the microchip. We perform reverse transcription, cDNA amplification, and library construction. And in the end, we have a sequencing-ready NGS library, which is compatible with the Illumina instruments. So let me now explain what exactly are the sequences that are attached to the barcoding beads. Every barcoding bead carries millions and millions of these sequences. So let's start from the right side. In green, you can see the poly-T sequence, which serves for the capture of the polyadenylated mRNAs from the cells. Then there is a blue sequence called UMI, which stands for a unique molecular identifier. Basically, every single oligonucleotide attached to the bead has a different UMI. And this UMI serves later during bioinformatic analysis as a quality control measure that helps you to determine how many of the mRNAs were actually captured in the individual microville. So it enables you to determine how efficient the mRNA capture in each microville actually was. The third important sequence is in orange, the cell barcode. So all the oligonucleotides that are attached to the same barcoding bead carry the same cell barcode, but the cell barcodes between two different beads are different. This helps us later doing bioinformatic analysis to assign all the transcripts with the same cell barcode to the same cell, because we know that they originated from the same cell. And basically, it helps us to distinguish which transcripts belong to which cell. And the last sequence that you can find on our barcoding bead is the barcoding beads is a PCR primer, which serves for the cDNA amplification and the library construction. So let me explain how this single cell RNA-seq experiments actually work and what exactly needs to be done to get the data. Our workflow is called JAXscope because our product, the kit that we are using in this workflow, is also called JAXscope. So our JAXscope single cell RNA-seq workflow starts with a tissue that can come either from the needle biopsy or from the surgical resection. This tissue gets dissociated into the individual single cell suspension. This suspension is loaded onto the microchip, where basically all the magic happens. So the cells get partitioned, they get lysed, and the mRNA gets captured. After that, we proceed with a reverse transcription, cDNA amplification, library construction, and sequencing-ready library is sequenced. And lastly, we perform the bioinformatic analysis. So for the handling of the pieces of tissue that basically we start with, we have two solutions called tissue preservation and tissue dissociation reagents. So the tissue preservation buffer can effectively preserve tissues for up to 72 hours at 4 degrees. We know that the tissues that are stored in this preservation buffer have minimally affected cell viability. So if we compare cell viability in the fresh tissues and the tissues stored in this preservation buffer for 24 and 72 hours, we can see that this viability remains largely unchanged. And also when we look at the gene expression data in this picture here in the middle, we can see that the blue dots correspond to the fresh tissue, red dots to the tissue stored in the tissue preservation buffer for 24 hours, and the green dots for tissue stored in our preservation buffer for 72 hours. And we can see that the pattern of these three colors remains largely the same, suggesting that there is no significant change in the gene expression pattern once you stored this, your tissues in our preservation buffer for up to 72 hours. So once your tissue is safely stored and transported wherever it has to be transported, you can continue with dissociation. For this step, we offer also specialized master mix, which enables you to achieve single cell suspension with high viability. If we compare our dissociation master mix with other commercially available master mixes or with the master mix reported in the scientific literature, we can see that the cell viability achieved using our SCell Live tissue dissociation master mix is much, much higher. Now, for the dissociation itself, you don't need any specialized equipment, but for those customers who prefer to have some level of automation, we offer Python instrument, which merges together a heating block and a grinding movements of a specialized dissociation tube, and enables you to process up to 8 samples simultaneously, providing basically higher level of automation for your dissociation procedure. All right, so we have covered what happens to the tissue, and now we are at the stage when we have single cell suspension. I should also mention here that for samples such as blood samples or the PBMC, you of course don't have to do any dissociation, and you can use these samples as well. They are compatible with our JAK-scope single cell RNA-seq workflow. So once you have your single cell suspension, you load it onto our microchip. And our microchip offers you another great advantage, which is that you can simply visualize under the microscope how your loading of the chip actually looks like. So on the left side, you can see our individual microvels that are empty. In the middle part of this green images, you can see individual cells occupying microvels on the chip. And on the picture on the right, you can see the single cells as well as barcoding beads occupying individual microvels. So once this partitioning of the cells and barcoding beads happens, the cell lysis takes place and the mRNA is being captured on the barcoding beads. The whole process, whole operation of the chip can be done without any specialized equipment. So as I mentioned, you only need a 20 microliter pipette to operate this chip. For the customers that require some level of automation, we offer a matrix instrument which is capable of processing 2 chips simultaneously within approximately 30 minutes. So once we have mRNA captured onto the barcoding beads, we proceed with the reverse transcription, cDNA amplification, and the library construction. And then the samples are sequenced. The library that we produce is compatible with the Illumina instruments. And lastly, we proceed with the bioinformatic analysis. So for the bioinformatic analysis, we have 2 solutions. One is called a Cellscope software, and the other one is called Synecosys Annotation Database. And I will explain in more detail what they actually do. So the Cellscope software is a freely available software that you can download from GitHub. The Cellscope software takes as an input raw data that the Illumina machine generates. And from this raw data, Cellscope software creates expression matrix. So by expression matrix, I mean a huge table filled with data. So for every single cell, you get the information on gene expression for every single protein encoding gene, which is actually in the organism that you study. So for instance, for human samples, you would get information on more than 20,000 protein encoding genes. For every single cell that is in your that you process in your experiment. So this would be thousands or tens of thousands of cells that you process simultaneously in a single experiment, and you get the gene expression information for every single gene for all of them. So this is a huge amount of data. And if you are thinking about how to visualize this huge amount of data, of course, it has to go through some kind of dimensionality reduction. And this is why we do so-called data clustering and t-SNE or UMAP-based dimensionality reduction. So in the single cell sequencing field, you see a lot of plots like this. These plots show clustering of the cells. And basically every single dot in this plot represents a single cell. So the cells that are close together, that are plotted close together, have similar kind of gene expression, and the cells with different kind of gene expression are plotted further apart. So once this clustering is done, the next step that usually takes place is the cell type annotation. So for this step, we offer a Synecosis database, which enables you to assign to every single cluster of the cells that you get using the Cellscope software. It enables you to assign to these clusters cellular identity. So it basically helps you to determine, helps you to identify, or helps you to assign microgenes to the individual clusters and helps you to identify them. So for instance, using the Synecosis database in this data set that I'm showing you, we were able to show that the red cells are the cancer cells, this green cluster are the fibroblasts, this brown cluster would be myeloid cells. So basically assignment of marker genes to the individual clusters is one of the main functions of the Synecosis database that we are offering. In addition to that, we also offer secondary analysis using Synecosis database, depending on what kind of research questions you are trying to answer. So for this whole bioinformatic pipeline, I have to say that it's your option whether you decide to do it yourself or whether you would like us to process your data. So basically we offer two different options. So the first one is basically do it yourself approach. We offer a kit which you can easily with which you can easily get single cell sequencing established in your lab. So as an input, this kit takes either tissue or the single cell suspension. And as an output, this kit gives you sequencing ready NGS library. In this situation, you can use our bioinformatic tools to basically do the analysis and the data visualization. The other option for the customers who prefer to outsource the whole single cell sequencing workflow, we have them covered as well. We offer a single cell RNA-seq service. In such case, you simply ship us your samples and we take care of the rest. So basically we perform all the steps in this workflow, including the bioinformatic analysis. And we basically provide you with the bioinformatic report in the end. So the whole process is very simple. You simply ship us your sample in our tissue preservation buffer. You get informed about all the steps that we do with your tissue and with your samples. And then in the end, you get your data in a publication-ready format. Also, I have to mention here that if you require any specialized kind of bioinformatic analysis, we have in-house bioinformaticians that would be willing to help you to accommodate whatever wishes you might have. So our single cell RNA sequencing service has so far served more than 3,000 different research projects. We've been working with more than 500 different customer organizations. And as an interesting fact, I should mention that we have sequenced more than 170 million individual cells. These cells come from either tissues or PBMC or different cell cultures. And we focus on the human and mouse data. So we have processed more than 400 different sample types, and our overall success rate is at 94%. And this is an example of how you can ship us your samples in case that you would be interested in our single cell sequencing service. We will send you a tube with the tissue preservation buffer in which you can simply drop your tissue. It should be around between 100 to 200 milligrams of the tissue per sample. And basically, we take care of the rest and provide you with the bioinformatic report in the end. So let me briefly talk about the applications of the single-cell RNA-seq that might be relevant for clinics, but not only. So we offer basically 4 different approaches of what you can do with our products. So first of all would be mRNA profiling. We offer single-cell RNA sequencing as well as single nucleus RNA sequencing. So for majority of samples, we recommend single-cell RNA-seq simply because you have more material and more mRNA in the single cells than you have in the nuclei. But single nucleus may be especially beneficial for instance, for frozen samples. So if you, for example, after this talk realize that you have some frozen tissues in your fridge or freezer, and you would like to start with single-cell RNA sequencing, these samples would still be eligible for the single nucleus RNA-seq. And here I'm just showing you some data which compares the performance of the single-cell RNA-seq versus single nucleus RNA-seq. This data comes from brain tissue. And basically the main message is that if you work with frozen tissue or with tissue such as brain tissue that is difficult to associate into individual cells, then the single nucleus RNA-seq should be the method of your choice. So the next group of products that we offer is called immune VDJ profiling. VDJ sequences are important for those of you who work with immune cells. So basically they define the specificity of the VDJ sequences define specificity of the T cell and B cell receptors. And for immunologists, it's especially important to understand or to get the sequence of the so-called CDR3 region in the VDJ sequences. And using traditional single-cell RNA-seq, this is difficult because we get a high gene coverage at the 3' end of the sequences, of the transcripts. But if we move further away from the 3' end, this coverage decreases. And for this reason, for the immunologists who want to study the CDR3 region, we offer a specialized kit that enables you to get sufficient coverage also of your CDR3 region that is further away from the 3' end of the transcript. So how it works is that we have specialized beads that this time not only have poly-T sequences for the mRNA capture, but they also carry specific probes on them. And these probes can capture the constant region of the T cell and the B cell receptor sequences, such that already at the first stage at the mRNA capture, we get enrichment of these sequences of interest of the T cell and B cell receptor sequences on the magnetic beads. Also later during the library generation steps, we perform 2 rounds of PCR enrichment for these VDJ sequences to ensure that in the end, you not only get the transcriptome information that you capture on the poly-T sequences, but you also get a sufficient coverage of the VDJ sequences around the CDR3 region that might be of your interest. The third group of products I would like to talk about are called focuscope products. They serve for the detection of genetic variants, fusion genes, and viral sequences. So basically the purpose of these products is very similar to the purpose of these VDJ detection kits that I just talked about. So we have a very high coverage at the 3' end of the transcripts, But if you are interested in mutations or gene fusions that are further away from this 3' region, you basically have to use an additional product, that enables you to get sufficient coverage there. And this is exactly what the focus scope is for. So with focus scope, you can select also customized gene panels of 5 to 8 genes. You can have specific probes on your beads, and you can get sufficient coverage of the sequences of your interest that you basically choose for your experiment. So let me show you how an example of the data from the focus scope kit. On the left side, you can see clustering of the cells coming from the lung tumor tissue. So basically, we performed a single cell sequencing experiment, and the individual cells, individual dots correspond to individual cells. The cells with a similar gene expression cluster together, and cells with a different kind of gene expression are plotted further apart. So this is what you see on the left part of the image, and the colors correspond to the individual cell types. So different cell types are labeled in different colors. On the right side of the image, we are looking at the very same data set, but this information here is, so to say, additional. So we were able to detect EGFR mutation positive cells in red. We plotted them in red. And basically, if you compare the two graphs, you can see that these EGFR mutation positive cells are primarily cancer cells. So basically the result that you would expect. So for those of you who study lung cancer, we have good news. We have already a kit available for the druggable mutation panel in case that you work on the lung cancer. We are working on other panels, for instance, on the breast cancer panel and the blood cancer panel. But also I want to emphasize here that you can always select your customized panel of 5 to 8 genes, and it's still possible to get sufficient coverage and to detect mutation of your interest in the genes that you would be interested in. So the customized option is still there. And the last group of products I would like to talk about are the products for detection of nascent RNA. So as I already mentioned, our single cell sequencing products enable you to get expression matrix. So basically gene expression data for every single gene in every single cell. And we are talking about thousands or tens of thousands of cells in this expression matrix. And even though this is a huge amount of data, sometimes it might not be enough, and you might require temporal resolution added to this kind of data. So an example when you would like to have that is, for instance, if you want to see if your drug, for instance, triggers any changes in the gene expression of your cell culture or in the experimental animal. So in such cases, we offer a first commercially available kit for detection of nascent RNA. It's called the Dynascope. And how it works is that we have a reagent, which is a cytosine analog. And we can inject this reagent, or you can inject this reagent in the experimental animal or add it to the cell culture. And from this moment on, all the newly synthesized mRNAs will have this analog incorporated into their mRNA. So basically later, during the cDNA transcription or cDNA synthesis process, you will get a mismatch in your sequence. And you will be able later bioinformatically detect what proportion of mRNA actually has this mismatch. And the higher the portion of mismatched mRNA, it means that the higher the portion of newly synthesized mRNA you are getting in your cells. So I just want to briefly show you the data, the kind of data that you can get using this temporal or using this metabolic labeling of mRNAs. So the data here you are looking at is again the single cell sequencing data clustered based on the similarity of their gene expression. So individual dots are the cells. If they are close together, they have similar kind of gene expression. If they are further apart, their gene expression differs. So in addition, we have color scheme here. So all the cells that have dark pink color have a high number of newly synthesized mRNAs. The cells with the gray color, they don't have that much of the newly synthesized mRNA. They, so to say, reuse the old mRNA that was already present in these cells before the treatment with our reagent. So using this temporally resolved data sets, you can answer different questions. For instance, you can take a look at the different kind of cell types, such as here, for instance, the B cells and the fibroblast. And you can see for individual populations of cells, how much of the new RNA these cell types actually synthesized during this time window that you were looking at. So for example, here in fibroblasts, you can see that almost no new synthesis of mRNA was taking place, whereas the situation in B cells is much different, and there is a much higher, the fraction of newly synthesized RNA per gene is much higher in these cells. And once you have this data, you can, of course, play around with them and plot them in different ways. For instance, on this lower plot here, I'm showing you a situation where we looked at the individual genes. So we took a look at the 6 genes, and for each of these genes, we plotted new transcripts in green and old transcripts in yellow. And we separated the expression of these new and old transcripts across 3 different cell populations. So the epithelial cells, fibroblasts, and the B cells. And for instance, for the gene Pilgricht here, we can see that the epithelial cells and the B cells were synthesizing quite a lot of new transcripts, whereas the fibroblasts were basically surviving with the old transcripts for this gene. So this is an example of what kind of data you can get using our products. Now I would like to briefly walk you through the case studies and through the publications that have successfully used our products in the past. So let me first talk about the paper published in Nature Communications. It's a paper on advanced osteosarcoma. So if you have ever worked with bone cancer, you probably know that it's an extremely difficult tissue for dissociation. And the authors of this paper had troubles finding the proper dissociation solution that would enable them to get single cell suspension and basically proceed with the single cell RNA sequencing. Unless, of course, they came across our JEXscope solution, which enabled them to basically sequence more than 10,000 individual cells, 110,000 individual cells. And in the end, the study was capable of providing potential therapeutic biomarkers. I would also like to mention here that the authors of this paper found high expression of the TIGIT receptor. So it's an immunoreceptor inhibitory checkpoint marker. And they found, the authors of the paper, that blocking of TIGIT enhances cytotoxicity of certain T cells against osteosarcoma. When this paper was published, we didn't know about just a few months later, after this paper was published, Roche announced successful phase 2 clinical trial that also used blocker of the TIGIT as an combinational immunotherapy. So I just want to mention here that using single cell RNA sequencing, you can get to clinically relevant results. And the second case study, and the last one I want to talk about, is about the advanced NSCLC. So this is a kind of lung carcinoma. So authors of this paper were able to sequence lung cancer samples from 42 advanced NSCLC patients. So advanced NSCLC is a condition that does not allow you to get a large amount of samples for sequencing. So basically the authors had to deal with small samples collected either by a needle biopsy or by bronchoscopy. So once the authors collected the samples, they stored them in our GEXscope tissue preservation solution. And using our GEXscope single cell RNA sequencing kit, they were able to get data of more than 90,000 individual cells. They were able to create cell atlas of the advanced single cell NSCLC. They were able to identify 11 major cell types, including the sub-cell types and the rare cells, such as, for instance, follicular dendritic cells. And lastly, the authors were able to plot the cellular composition for every single patient and every single sample, allowing them to get insights on the interplay between the immune system and the cancer cells within the samples. So I already introduced you to single-cell sequencing. I provided you an overview on the single-urns technology, and I briefly walked you through some of the published case studies that have successfully used our products. I just want to mention to you that if you were thinking about beginning with single-cell RNA sequencing, our service option might be just right for you because it's very it's simply to do from your side. You just send us your tissue and receive the bioinformatic analysis in the end. So basically, it's very customer friendly from my perspective. And last thing that I want to mention is our special offer that we have running until the end of March. So we offer free single-cell RNA-seq library preparation for those customers who challenge us and who provide us with human tissues, fresh human tissues that we haven't processed in the past. So as I mentioned, we process more than 400 different tissue types. But for the first 3 customers that happen to provide us with a tissue that we haven't touched before, those customers can get free single-cell RNA-seq library prep. And with this, I want to thank you all for your attention. I would be happy to get your questions and answer them. So please just write them either in the chat, or you might be able to raise a hand and also talk to me in person. Or if you decide to have some questions, not now, but in the future, feel free to reach out and contact us either via our email form or social media. Thank you very much for your attention.
Yes, thank you, Dr. Pankova, for the very interesting talk. So I think we could start the Q&A section, and we already have some interesting questions, and I would like to have this webinar as more interactive as possible. So please feel free and ask your questions. So the first question is that, is there any difference in cell capture efficiency and performance between manual and automated loading using matrix instruments?
So far, to my knowledge, there is none. The one difference that I am aware of is the initial concentration that you begin with when you want to load your single cell suspension onto the chip, either using the manual method or the matrix. So if you want to use less concentrated single cell suspension, then the manual loading allows you more freedom to basically, you don't have to concentrate your single cell suspension as much as you would have to do it for the matrix loading. But other than that, I don't think there are any notable differences.
If you also would like to discuss further, you can use the raise hand button and discuss further. So And the next question is that how many cells can be processed in a single run in a manual way?
Yes. So for single run, basically for the manual as well as for the automated version, there is no difference. We have two different, so to say, options for you on the throughput of a single experiment. So we have one kind of chip, which we call standard chip. This chip has 150,000 microvels, and we use it, or we recommend we recommend capturing of up to 10,000 individual cells on this microchip. The other option is so-called high density microchip. High density microchip has 250,000 microvels present on this chip, and you can capture up to 30,000 cells in this high density configuration. But again, there is no difference between the manual loading and the loading used in our matrix instrument. So the capturing efficiency should be the same.
And next one, how long does a single run service take from samples to ready data analysis?
So currently, I think it takes around 7 weeks. So this is the time between the point when we get your samples, and in 7 weeks you can expect from us the complete bioinformatic report with the data analyzed and visualized.
If the user is interested in only TCR sequencing, would you recommend VDJ TCR-PCR or a circle TCR full length? Good question.
So for the VDJ sequencing, you have always a possibility to only create library containing either TCR or the BCR receptors. So our kit is designed such that you can amplify both of them. But if for whatever reason you prefer to only have one of this population, the VDJ kit allows you to do that. So basically you can still only choose to amplify that population of TCR or BCR receptors that you are interested in.
Yes, thank you. So next one, in-person success rate. May I know what's the cause of failure of those single-cell RNA samples that didn't pass? So, for example, not able to process because of low cell viability, or the single-cell RNA-seq results weren't good because of few UMIs or genes.
Yes, so the tissue dissociation is the basically first and most important step in your single-cell experiments. And even though we have a very good success and overall success rate in processing different kind of tissues, there are some kind of tissues that might be problematic, especially the ones which we work for the very first time. But I have to say that at the moment, with our experience in more than 400 different tissue types that have already been processed, there is a high chance that the tissue that you are working on, you already have some experience with.
Mm-hmm. So I have also a question regarding the preservation buffer. So what is the exact tissue treatment process? And so also we have another question asking about for me, it is difficult to reach 100 milligram of tissue, the amount of basically tissue. Would single run still accept preserved tissue samples with lower weight? What is the minimum cell number expected from the preserved tissue sample?
Yes, so in general, we recommend that the amount of tissue that you send to us is between 100 and 200 milligrams. However, sometimes we accept also the lower amount of tissues. But the customer should be aware that this is kind of a risky zone. So basically, the customer should be aware that we cannot guarantee successfully getting good results with such low amounts of tissue. But I have to say that, for instance, in the picture that I have been showing you on the nasal biopsy here that has been sent to us, in this case, the weight was not even measurable, and we were still able to successfully dissociate this tissue and prepare the library and get a single cell RNA-seq data from it. So yeah, our recommendation is about 100 milligrams, but lower amounts are possible, but however, we cannot guarantee that it will work out.
And how about the process?
Yes, so the whole process of the tissue, how you send us the tissue, it will be discussed in the next webinar in 1 week in more detail. So we have a technical expert that will describe in more detail what are the requirements of the sample, what kind of samples we do and do not accept, what are the quality control steps, and so forth. But basically, the procedure for you is rather simple. We send you a tissue preservation buffer, a box with tissue preservation buffer. You just drop in your tissue in there, and then your part is done. You just wait for us to provide you with the quality control steps. We keep you informed along the way as we process the tissues. And in the end, you get your bioinformatic report. Yes.
And which tissues are suitable for this preservation buffer?
So for this tissue preservation buffer, as I mentioned, we have experience with more than 400 different tissue types. So the tissue preservation buffer itself is a rather simple solution made of salts and some vitamins and glucose. So basically it aims to serve as a physiological solution that keeps the tissues in a good condition and doesn't trigger any gene expression changes in them.
So how long does it take for these? Oh, okay, so I asked this question already. Yeah, I think we answered this question. And yeah, I think we're done with the questions. And thank you very much for attending. If you have any other question, just feel free to reach out. After this webinar, we will be glad to answer any questions of yours. Thank you so much and have a great day. Bye.
Thank you. Bye.